So our second speaker is Dr. Ruth Marfil Vega, who's the environmental scientist in the Innovation and Environmental Stewardship Program at American Water's Central Laboratory in Belleville, Illinois. She's leading research there focused on emerging contaminants, and she received her PhD at the University of Cincinnati in environmental engineering. Her talk today, she will be presenting an overview of American Water's Innovation and Environmental Stewardship Group and its studies on the fate of emerging contaminants. So with that, I'll turn over the presentation to you, Ruth.
Thank you, Nancy. Can you see my presentation now? Can you hear me well?
We can hear you just fine, and you just need to go to the slideshow, and we'll be ready to go.
I have the slideshow up right now. It's not. Give me just 1 second.
Switch screens if you have a second screen.
Yeah, that's the problem. Can you see it now?
There it is. Yep, perfect.
Perfect. Thank you so much. Thank you, Nancy, for inviting me to talk about the emerging contaminants or emerging constituents program that we are developing at American Water, and also thank you, Helen, for the great overview about what these chemicals are, and put everything a little bit into context, because sometimes we forget that one of the key aspects is to understand the risk that these chemicals might have. And as I just mentioned, I'm going to be talking about emerging constituents. If you pay attention to my long title, I change. I normally talk about emerging contaminants, but one of the outcomes that of a research project that we supported with the Water Research Foundation, looking into improving terminology to communicate about these chemicals, was that the constituents is a more neutral word that should be used when communicating with people outside the scientific field regarding the emerging chemicals or constituents of emerging concern. Today I'm going to be providing an overview of what American Water is, for those who don't know it, what are our research interests, and I'm going to have a series of very busy slides, because I was just trying to give a snapshot of the research that we are doing, and whenever show you some of the results that we are getting. You know, for people from Illinois, you might be more familiar with Illinois American Water, but for those that live outside there or don't get the water from the company, I just want to say that American Water is the largest publicly traded water provider in the U.S. We run and operate drinking water treatment plants, wastewater treatment plants, desalination plant, and reclamation plants. So overall, we serve water to 16 million people all over the country. Within the company, we have an innovation and environmental stewardship group that is formed by over 20 people. We have four different groups that collaborate very closely between each other. One group is the environmental compliance and stewardship. The second one is the central laboratory, that is where I'm located in Belleville, in southern Illinois, and you can see a picture down there. And this lab focuses on performing all the analyses that are required by regulation for the drinking water operations. Then we have the research group, and another group that looks into the innovation development program, looking into new technologies that can be applied in the drinking and wastewater treatment. The water industry is facing a lot of challenges currently, and highlighted in red are the ones that are more relevant to the topic of the presentation today. Those are the emerging constituents, new environmental regulations, and also environmental concerns. People are more aware of things in the water that might might be harmful or might be risky. It's very easy to get information from internet that is not necessarily accurate or scientifically sound. So that's one of the areas that we also focus. The more specific goals of the research program is to address emerging water quality issues and as well as anticipate regulation that may come up at state or federal level. Also, we do a lot of work on evaluating new technologies that will allow us to improve operations for current regulated compounds, but as well as improve new technologies for the treatment of chemicals that might be regulated in the future. And we also provide, the research group provides support to the operations of the drinking and wastewater treatment plant. Our communication department reaches to us quite often when there are news appearing in magazines of generic interest to ask us if that's accurate. And overall, we provide the support to the whole company. Some of the topics that we are of interest to us right now are disinfection by products. When the DBPs, THMs, and HAAs were regulated a few years back, a lot of drinking water treatment plants switched from disinfecting with chlorine to use chloramines. And when that happened, a new group of chemicals started appearing in the water, and those are the disinfection byproducts resulting from chloramination. For the past 3 years or 5 years, we've been doing a lot of work on biofiltration. During the drinking water, as well as in some cases during wastewater treatment, there is a step in which the water is filtered by a carbon filter, similarly to the Brita filters that you might have at home. Though that carbon is replaced every 3-5 years or regenerated, but that has an associated cost. And what we've been looking in the past years is to the impact if instead of replacing the filters on a regular basis, we replace the filters based on performance. If we let the media in the filter for more than 3-5 years, what's going to happen? And if the media is left there, we are also encourage bacteria to grow on the surface of the media, so we can move from a removal mechanism that is based on absorption to a mechanism that is going to be based on biological activity. We've also been working for a long time on algae blooms. EPA released this summer the health advisory for some cyanotoxins, but even before that, the company has been using a new technology based on ultrasound that destroys it doesn't destroy the algae, so the toxins are not released, but what it does is it alters the oxygen content within the algae, so the algae sink to the bottom, and then when the algae start dying slowly in the bottom of the reservoir or the lake, the toxins are released at a very slow rate. So in this case, we don't run into problems with the toxins, the cyanotoxins or cylindrospermopsin being above the health advisory that was released by EPA this summer. This is a technology that was developed in the Netherlands, and we implemented in some reservoirs in New Jersey 3 summers ago, and now we are expanding the installation of these devices in different utilities. It was mentioned earlier also the risk and the presence of microbial contaminants. We've been doing a lot of work in the Legionella, and also we are expanding now to work on other emerging pathogens, like for example Naegleria. As you might have heard, there are more cases of people dying because of the presence of Naegleria in the distribution system in the past years. And finally, we are also working or devoting a lot of effort to evaluate pharmaceuticals, personal care products, and other emerging constituents. As I mentioned previously, we operate on drinking water treatment plants, but also wastewater and reuse plants. So basically, this gives us ownership of the full water cycle. So we are looking into understanding the fate and the movement of these chemicals going from one of the treatment steps to the next one. We normally tend to think about drinking water or wastewater treatment as a linear process, and we sometimes forget that they are all interconnected. And I'm gonna show some of the results of projects that we've been working in the past years, and some of the new ones that are coming up. Nitrosamines. Why are we worrying about nitrosamines? These chemicals meet one of the three problems that I mentioned earlier the water industry is facing. Nitrosamines are carcinogenic. They are going to be most likely regulated by EPA in the near future at a level of 10 nanograms per liter. And we don't really know much about the formation and removal besides that the nitrosamines form when you add chloramines into the water. This is a very complex slide, but the summary, the key message is like organic matter is going to get into the drinking water treatment plant and is going to be a precursor of disinfection byproducts when you disinfect the water. The nitrosamines are going to be forming during the drinking water treatment process. But also on the other end, on the right side of the slide, the wastewater treatment plants are going to be releasing different type of organic matter and again precursors, but also nitrosamines are going to be forming there. And then if there is a reuse happening right after that, that's going to increase the risk of formation again of nitrosamines in the drinking water. In one of the projects that we conducted in partnership with the Metropolitan Water District of Southern California and the University of Alberta, we monitored 40 drinking water treatment plants in the US and Canada. And one of the situations we ran into is what I'm showing on top. You can see that the nitrosamines, the NDMA formation increased during treatment, and this was caused by the addition of different coagulants and polymers that are added during the drinking water treatment, but especially in the distribution system, the sampling locations that are shown at the end of the top plot. Those results demonstrate that when chloramines were added to the water, there was a large formation of NDMA reaching 50 nanograms per liter. We ran into a different situation, and that's what is represented in the bottom slide. We have NDMA presence at around 10 nanograms per liter, so the question was, what's going to happen if we change the operations in this drinking water treatment plant? Are we going to go above the 10 nanograms per liter? And something that we can use to estimate the amount of precursors that are present in the raw water coming into the plant is the use of artificial sweeteners. The artificial sweeteners are like, for example, sucralose that is found in Splenda, acesulfame, and these chemicals don't get degraded during wastewater treatment plants. So when you intake these compounds, your body releases them, they reach the wastewater treatment plant, and because the technology is not efficient to remove them, they get into the surface water and then go to the drinking water. So we found that analyzing for this sucralose and acesulfame K, we can find a relationship between the precursors of nitrosamines that are coming from raw waste, from the wastewater that is getting into the source. Another source of nitrosamines precursors can be wastewater treatment, and we performed this project that we are just finishing, looking at improving operations by using lower amounts of oxygen during the treatment. And the main goal was to reduce greenhouse gases as well as reduce the cost of pumping oxygen in the wastewater treatment plant. So what we did, we monitored NDMA, that is it's plotted in blue, in the effluent of this wastewater treatment plant, and at the same time we've also run a test to determine the total amount of precursors. But we wanted to know from that total amount of precursors what's the role of emerging contaminants. Our interest in this was driven by the fact that there's been several publications indicating that some emerging constituents can result in the formation of NDMA if they are disinfected with chloramines. For this work, we've been working on developing the artificial sweeteners method, as well as some emerging contaminants. And because we are using an LC QTOF, we have the capability of doing the targeted screening, and you can see in the blue box some of the chemicals that we targeted for this specific project, but also we are able to do some untargeted screening. This slide summarizes the results that we got in this effort. We were able to quantify sucralose and acesulfame in the effluent. Carbamazepine and DID were also quantified in the effluent, and you can see that the yellow boxes contain different chemicals with a question mark. The way we identified those chemicals was by using the screening capability of the QTOF, and once we ran the samples and we quantitated the compounds that were targeted, we were able to process acquisition the results. And we have a library that contains over 2,000 chemicals, and we just look for 135 chemicals in negative mode and another 100 chemicals in positive mode. And we employed some filters that are, for those more interested in the technical aspects, are listed at the bottom. And with that approach is how we identify the chemicals that were included in the yellow box. And what is interesting from the nitrosamines study point of view is that all those chemicals contain nitrogen as the nitrosamine. Some examples of projects looking at the implementation of biofiltration is this one that we are partnering with the Southern Nevada Water Authority as well as the University of Colorado and Carollo Engineers. The tasks that the American Water is conducting is looking at atrazine in two different filters. One of them is a new filter where the media was replaced in May, while the other one is a filter that's been in place in the plant for over 2 years. So we started the monitoring in April before the planting season because we wanted to capture the spike of atrazine in the source where this plant is taking the water. And on the left side of the slide, you can see the results from April to August that we found for the atrazine. Filter 1 is the old filter, while filter 6 is the new one. So filter 6 is after 5 months is showing a better performance for the removal of atrazine. But this effort is continuing, and we'll see the full picture of the behavior of the filter till April that we complete the monitoring. As I said, the filter performance was better, but this year the concentration of atrazine that was found in the river was well below the 0.3 milligrams the 3 micrograms per liter MCL for atrazine. Additionally, because we are using the QTOF for this effort, we are able to monitor other chemicals that initially we didn't know about it, and those are the different transformation products from atrazine. These transformation products, like hydroxyatrazine and the others that are listed on the right side, are formed during the soil by bacterial activity in the soils. So these compounds are getting also into the source water, and then we are picking them up during the drinking water treatment. So we are still doing a lot of effort to evaluate this data and also look into other compounds that were not targeted initially in the method. One project that started a couple of months ago, and today we are receiving the first set of samples, is this additional project. This additional project looking at the use of biofiltration. Water reuse is becoming a key technology in communities affected by the water scarcity, and California is the state that is driving the regulation. And in California, if you want to have a reuse project, you need to use what is called FAT technologies, like it's full advanced technology that includes membranes, RO, reverse osmosis, advanced oxidation, and it's very it requires a lot of energy. And also it requires that you need to dispose the reverse osmosis concentrate. So we believe that this is not the best alternative for communities that are not near the ocean, and we are looking at the efficiency of ozone biologically active filters as an alternative to this FAT approach that is implemented in California. As I mentioned, we are starting to get samples today, and we are doing a survey of several facilities all across the country, and we are going to be setting up a pilot plant in which we are going to be evaluating the fate of the chemicals that are listed on the table. And as you can see there, many of them are matching the compounds that were found in Minnesota and the compounds that the risk assessments were completed, as Helen mentioned earlier. We are doing some research on membranes, also to apply them to desalination plants and as well as reuse. We've been monitoring. We have this project to evaluate new chemicals that can be applied to minimize fouling of the membranes. And we've been monitoring 6 locations, 3 wastewater treatment plants, as well as 3 desalination intakes for desalination plants. And we've been evaluating the efficiency of these new materials. And at the same time that we evaluate the materials for the removal of traditional water quality parameters, we've been monitoring emerging constituents. We've been lucky that we haven't been finding a lot of these chemicals. The frequency of detection is low, as well as the concentrations are very variable. Something that we can use the results from this project is also for the outreach the company does. Like, for example, in the plots that are included here, we are showing the results from DEET, and we can see that between the two different locations, the concentration of DEET is quite different, even though both sets of samples were collected during the summer. So we can target for people near the desalination number 1. It seems that the usage and the hydrology of the location where the sample was collected is it's higher, or the movement of the DEET in the ocean is lower. So we can approach different outreach strategies to educate people about how to minimize the presence of emerging constituents in the environment. So overall, our effort is somehow focused on changing the messages that appear in these cartoons from time to time, and try to get to a point in which the water that we have, it's safe for humans as well as the ecosystems. I need to acknowledge my team here at the Central Lab, as well as the rest of the people in New Jersey, the agencies that are funding most of the work. And with this, I'll be happy to answer any questions.
Okay, thank you very much, Ruth, for an excellent talk about your program. I'll now see if we have any questions here from the audience, and then we'll relay those to you. Are there questions anyone would like to ask?
Yeah, I had a question on the biofiltration results. I just couldn't tell, you had a filter 1 and a filter 6. Were both of those biofilters, or was one a non-biofilter and the other one a biofilter, just to understand what the impact of the biofiltration was compared to a non-biofilter?
In April, when we started the monitoring, both of them were biofilters. Then the filter number 6 was replaced. We collected we are taking April as the reference point, also that sampling happened before atrazine was applied in the field. So in May we can see the impact, the increase of atrazine in the river, and then is when you can see the difference in the removal by the filter between number 1, that is the biofilter, and number 6, that is the new media. The first, we have the 2 influence and the 2 effluents plotted side by side.
Okay, thank you. Anything else? Just a moment.
Probably my fault because I'm sitting way in the back, but one of your later slides you mentioned, or you had on there, heated metal oxide particles.
Mm-hmm.
So are you heating the particles to use adsorbents, or are you heating the water that the particles are in?
This is a project that Mark Benjamin from the University of Washington patented this process, and these are iron and aluminum particles that are heated and are synthesized. This work is being done in the lab in New Jersey, so I'm not very familiar how the particles are prepared. But these are the iron or aluminum are heated, and these new particles are made. And in the project we are comparing them with alum and ferric chloride, to have like the traditional particles that could be most similar to these new ones.
Is there a publication on that work that's out yet, on the heated metal oxide research?
Mark Benjamin had some publications. This work that I presented today is work in progress, and we don't have the publications out there. But on the process of preparing them, it should be published.
Okay, thank you. Anything else here? With that, we want to thank you again, Ruth, and we'll just, if anyone has questions, they can email you or call you, and then we're going to just turn it over to some discussion here amongst our attendees to see if there's different ideas they have or not. So thank you very again, Ruth.
Thank you.